Imagine a world where we don't just, understand life, but we can actually redesign it, even build biological systems totally from scratch.
Right. What if we could program life kind of like we program a computer?
Exactly, to solve huge challenges like creating brand new medicines or maybe cleaning up pollution.
That's really the core promise of synthetic biology, and it's moving fast, It's shifting from sci-fi into, well, into reality.
So it's not just tweaking existing genes.
Not entirely. While traditional genetic engineering often involves moving existing genes around, synthetic biology is more about designing and constructing new biological parts, devices, systems, sometimes building them right from the ground up.
Okay, so that's our mission for this deep dive then. We're going to explore synthetic biology, where it came from, the tools involved, and crucially, the huge impact it's already having.
And we definitely need to get into the big ethical questions too. This power, it raises some profound issues for society.
Absolutely. By the end, you should have a really clear picture of this new frontier and what it means for all of us. So let's jump in. Where do we start? The history.
Yeah, let's do that. To really get synthetic biology today, you need to look back. It didn't just appear out of nowhere. A key moment was way back in 1961.
1961. What happened then?
That's when Jacob and Monod published their work on something called the lac operon in E. coli.
The lac operon. Okay.
It was huge because it was the first time scientists really understood a gene regulation pattern. It showed gene expression wasn't just random, it's controlled, predictable, like a biological circuit.
Ah, okay. So it revealed a kind of underlying logic to life's code.
Exactly, like finding the first bit of the blueprint for how cells control themselves. And that understanding, well, it paved the way for what came next.
Which was?
The 70s and 80s. That's when we saw tools like molecular cloning and PCR developed. Suddenly, scientists could copy and paste DNA much more easily. Genetic manipulation became, widespread in labs.
Right. Standard practice in microbiology research.
Yeah.
But that was still mostly moving existing bits of DNA around, wasn't it?
Yeah.
When did the design aspect really kick in?
That shift really started to crystallize in the early 2000s. Things started moving towards engineering biology, not just studying it.
And a big part of that was Tom Knight at MIT with the biobricks, right?
Yeah.
Around 2003.
Absolutely. Biobrick plasmids. Think of them like Lego bricks, but for genes. Standardized biological parts.
So you could easily snap them together in different combinations.
Pretty much. They could be combined using standard methods, stored easily, and shared. It really changed how people thought about building genetic systems. There's even an open source foundation maintaining the standard now.
That idea of standard parts, modularity, sounds very engineering.
It totally is. And it enabled a key step. In 2000, two major papers came out in Nature. They reported the creation of the first synthetic biological circuits inside E. coli.
Actual circuits, like in electronics.
Kind of. One was a genetic toggle switch, like an on-off switch for the cell. The other was a biological clock, an oscillator. This wasn't just modifying existing pathways, it was building predictable behaviors from scratch.
Wow. Okay, so the field is building momentum. You've got the basic understanding, the tools, the standard parts, the first circuits. What came next?
Community. Collaboration. In 2004, the first IGM competition, that's the International Genetically Engineered Machine Competition, launched at MIT.
IGM, yeah, I've heard of that. It's huge now, isn't it?
It exploded, became this global phenomenon, bringing students and researchers together from biology, chemistry, engineering, computer science, all focused on designing and building biological systems. It really fueled the interdisciplinary nature of the field.
Okay, so the community's growing, the tools are getting better.
Yeah.
Then came the really big leap, right? Synthesizing whole genomes.
Yeah, moving beyond just editing bits and pieces to actually writing large stretches of DNA, sometimes totally new sequences, and putting them into organisms.
That sounds
It is. A major milestone, or maybe a warning shot for some, was in 2002 when scientists synthesized the entire poliovirus genome from scratch.
the poliovirus.
Yeah.
From scratch.
Yep. Chemically synthesized DNA based on the known sequence. It worked. It was infectious. Caused a big stir, obviously.
I can imagine. Suddenly, the worry wasn't just finding dangerous viruses in nature, but someone making them.
Precisely. That raised huge biosecurity flags right away. Then, moving beyond viruses, in 2008, came the first synthetic bacterial genome, Mycoplasma genitalium.
So writing the entire operating system for a bacterium.
In essence, yes. And work continued partial synthesis of the yeast genome in 2017, and even a bacterial genome designed entirely by computer in 2019, though it wasn't viable yet. It shows the direction things are heading.
Okay, so that brings us more or less up to date on the history. Let's talk about the modern toolbox. How does this stuff actually get done? But CRISPR has to be mentioned first, right?
Absolutely. CRISPR-Cas9, which really took off around 2013, was a revolution. It's like molecular scissors that you can program.
To cut DNA at a very specific spot.
Exactly. It locates the target sequence, makes a cut, and can even help insert or replace DNA there. It's just so much faster and easier than older methods, especially for complex cells like human cells. It democratized gene editing in a way.
But synthetic biology isn't just CRISPR, right? Those biobricks you mentioned are still key.
Definitely. That principle of modular design, creating parts that work predictably, like Legos, is still fundamental. You need reliable components to build complex systems. The Registry of Standard Biological Parts is a huge resource for this.
And these parts get assembled into genetic circuits. You mentioned the toggle switch and the clock earlier.
Right. Genetic circuits use combinations of genes, often responding to inputs like heat, light, or specific chemicals to produce a desired output.
Like programming logic into a cell.
That's a great way to put it. You can design A and D gates, or gates, not T gates, just like in electronics, but using biological components. This lets you create sophisticated behaviors like cells that only produce a drug when they detect two different disease markers, or cells that form patterns, or even that bacterial photography where E. coli could sense light.
It really is merging biology and engineering. But it's not always about designing from first principles, is it? What about directed evolution?
That's another really powerful tool. Instead of trying to perfectly design a protein or function, you basically use accelerated evolution.
How does that work?
You create many, many variations of a gene or protein, lots of small mutations, and then you screen them under high pressure for the ones that do what you want, maybe slightly better. Then you take the winners, mutate them again, screen again. It mimics natural selection, but on a much faster timescale.
So you can kind of evolve molecules to have newer improved functions?
Exactly. It's great for creating things nature hasn't produced, or making existing enzymes work better under industrial conditions, for instance.
And now AI is getting involved too.
Hugely. Computer science and AI are driving the modern era of synthetic biology. AI can help design incredibly complex metabolic pathways, figure out the best way to build intricate genetic circuits, even, as we mentioned, design whole genomes.
But that has its own challenges.
It does. The power is immense, but sometimes an AI might design something that works, but we don't fully understand why it works. If something goes wrong with an AI-designed organism, debugging it can be really tough. It can be a bit of a black box.
Okay, powerful tools, complex designs. Let's get into the applications. Where is synthetic biology actually making a difference right now? How about environmental solutions first?
Yeah, that's a really active area. Think about pollution. Scientists are engineering microbes as tiny biosensors.
Sensors, how?
They can be designed to detect specific toxins, say arsenic or mercury in water, and then produce a signal. Maybe they glow or change color. It's a quick way to flag contamination.
And can they clean it up too?
Sometimes, yes. Other microbes are being engineered to actually consume pollutants, like industrial chemicals, or to bind to heavy metals, effectively removing them from the environment, like a specialized microbial cleanup crew.
That's fascinating. And it goes beyond pollution, right? What about biodiversity? I heard something about de-extinction.
Right. It sounds like science fiction, but companies like Colossal Laboratories are seriously working on it. They're using synthetic biology tools like CRISPR to edit the genomes of living relatives.
Like editing elephant DNA to resemble a woolly mammoth.
Sort of, yeah. Or editing gray wolf cells to try and bring back traits of the dire wolf. The idea is to create proxies for these extinct species and eventually reintroduce them to help restore ecosystems. It's controversial, but technologically, well, they're pushing the boundaries.
Wow. And what about preventing extinction or controlling invasive species?
Also happening. Gene drives are a key technology here. These are genetic systems designed to spread rapidly through a population.
Spread a specific trait.
Exactly. For example, making invasive golden mussels infertile to control their numbers, or modifying mosquitoes so they can't transmit malaria. Lab tests on mosquitoes have been successful, though field trials are still a big step.
That sounds powerful, but also potentially risky if it goes wrong.
Absolutely. Gene drives require extreme caution and containment. But the potential benefits, say for malaria control or protecting native species, are huge. There's also work on making corals more resistant to heat stress or engineering soil microbes to help plants survive drought.
Okay, so environmental applications are pretty broad. What about food and agriculture? That feels like another huge area.
Definitely. Crop improvement is a big one. Engineering plants for drought resistance, like tomatoes, or promoting deeper root growth, which helps with stress and sequesters more carbon in the soil.
And reducing fertilizer use. I heard about microbes for that.
Yes, this is potentially game-changing. Companies like Pivot Bio are engineering microbes that live with crops like corn, wheat, and rice and pull nitrogen directly from the air, feeding it to the plant.
So the plant gets its own fertilizer factory right at its roots.
Essentially, yeah. It could drastically reduce the need for synthetic nitrogen fertilizers, which have a huge environmental footprint.
And then there's the food itself.
Hmm.
Alternative proteins, right? Like the Impossible Burger.
That's probably the most famous example. They use synthetic biology to produce heme conestor. It's an iron-containing molecule found in meat. Gives it that characteristic flavor and color. They get yeast to produce it using a plant gene.
And the result is a plant-based burger that tastes more like meat and has a much lower environmental impact.
Significantly lower. Less water, less land, fewer greenhouse gas emissions compared to conventional beef.
Mhm.
It really shows the potential. And other companies are using engineered microbes to produce other food proteins, mimicking dairy or other meats.
So better crops, less fertilizer, meat alternatives. What else?
Well, think about flavors and materials. Most synthetic vanillin on the market now, made by genetically modified yeast. That same yeast can also be engineered to produce spider silk proteins.
Spider silk from yeast.
Yep. For making strong, biodegradable fibers. Again, with less environmental impact than traditional methods. It shows the versatility yeast as a tiny factory for all sorts of complex molecules.
Okay, that leads nicely into innovating products and materials beyond food. What's happening there? Greener manufacturing.
For sure. Biofuels are a classic example, engineering microalgae to be more efficient at photosynthesis, capturing more carbon, or designing microbes that can ferment waste gases from factories into ethanol.
Waste gases into fuel.
Yeah, a company called LanzaTech has a plant in China doing just that, taking emissions from a steel mill and turning them into millions of gallons of ethanol, turning pollution into a product.
That's clever. What about plastics? Can synthetic biology help with the plastic problem?
There are efforts on both ends. Some researchers are engineering bacteria with enzymes that can actually break down PET plastic, the stuff in water bottles.
Bacteria that eat plastic.
Basically, yeah. And on the production side, you have companies using microorganisms to create bioplastics, sometimes from waste gases like methane or CO2, making materials for furniture or packaging that are biodegradable.
And even building materials. You mentioned bricks earlier.
Right, biomason. Using bacteria to essentially grow bricks from sand and calcium ions. It takes just days, uses no heat like traditional kilns, and they can even engineer properties like making them glow in the dark or absorb pollutants. It's rethinking construction.
Wild. And I saw something about DNA for electronics or data storage.
Yeah, that's pushing the envelope. DNA nanowires as potential alternatives to silicon. And DNA data storage using DNA's incredible density to store vast amounts of information for long periods. Companies like Iridia are working on DNA-based memory chips.
It sounds like the applications are just exploding everywhere. But maybe the most profound impact is in medicine and health.
Arguably, yes. Let's start with drug production. We mentioned yeast making vanillin and spider silk. The same principle applies to complex medicines.
Like things that are hard to make chemically.
Exactly. Microbes like E. coli or yeast are engineered at cell factories to produce things like artemisinin, a key anti-malarial drug, getting microbes to make it boosted yields significantly compared to extracting it from plants. Same goes for some anti-cancer drugs like paclitaxel, even precursors for opioids or cortisone.
So it makes making these drugs more reliable, maybe cheaper.
That's the goal, more scalable and sustainable production. There are even cell-free systems being developed now, using just the molecular machinery outside the cell for even more control.
Okay, production is one thing. What about diagnosis?
Huge advances there, too. Sequencing technology, next-gen sequencing, third-gen sequencing, lets us rapidly identify genetic mutations linked to diseases or identify unknown pathogens incredibly quickly. Think about tracking Ebola outbreaks in real time with portable sequencers like the Minion.
And those biosensors you mentioned for pollution, can they detect diseases?
Absolutely. Engineered cells or cell-free systems with specific genetic circuits can be designed to detect disease biomarkers, proteins, specific DNA sequences, chemicals with really high sensitivity and specificity. You can imagine wearable sensors constantly monitoring your health.
That leads us to treatment, which feels like the ultimate goal for many.
And where synthetic biology is incredibly active. We've already talked about CRISPR for editing genes. Beyond just cutting, there are now base editors that can precisely change a single DNA letter without breaking the DNA backbone.
Even more precise.
Yeah, potentially safer for certain applications. Showing promise for diseases like Duchenne muscular dystrophy or cystic fibrosis. People are even exploring editing multiple genes at once for complex diseases like cancer, or even trying to reverse aging markers in animal models.
And therapies based on RNA.
Right, programmable nucleic acids. Things like siRNA or antisense oligonucleotides, ASOs, can be designed to target specific messenger RNA molecules and silence disease-causing genes. The first FDA-approved siRNA drug, patisiran, came out in 2018.
Then there's cell therapy, like CAR-T.
CAR-Art is a fantastic example of synthetic biology in action. You take a patient's own immune cells, T cells, engineer them with a synthetic receptor, the chimeric antigen receptor, or CAR, so they specifically recognize and kill cancer cells?
And synthetic biology makes these CAR-T cells smarter.
Exactly. You can build in logic circuits. Maybe the T cell only activates if it detects two cancer markers, an A and D gate, making it more specific and reducing side effects. Or maybe it can be controlled externally. 5 CAR-T therapies are already FDA approved. It's saving lives.
Amazing. What else is in the treatment pipeline?
Lots. Engineered viruses are used as vectors to deliver therapeutic genes or gene editing tools. There are over 20 approved therapies using these. Phage therapy is making a comeback too.
Phages. Those are viruses that infect bacteria.
Yep. Genetically engineered phages are being developed to specifically target antibiotic-resistant bacteria. Huge potential for tackling the antimicrobial resistance crisis. There are dozens of clinical trials underway.
Bacteria fighting bacteria.
Or even bacteria fighting cancer. Scientists are engineering bacteria that specifically home in on the low oxygen environment inside tumors and release anti-cancer drugs right where they're needed.
And pig organs for human transplant. Is that synthetic biology too?
It heavily involves gene editing, which overlaps significantly.
Yeah.
Editing pig genomes, sometimes changing 10 or more genes to make their organs less likely to be rejected by the human immune system and safer regarding potential virus transmission. Xenotransplantation offers huge hope for organ shortages.
It really feels like we're entering a new era of medicine.
We are.
Uh-huh.
And tools like organ on a chip, tiny microfluidic devices that mimic human organ function, help test all these new drugs and therapies more effectively before they ever get near a patient.
Okay, the possibilities seem almost limitless. But with this much power comes responsibility, right? We have to talk about the unethical side, the societal implications.
Absolutely crucial. And maybe the first place to start is just clarifying that line again, genetic engineering versus synthetic biology. It's debated, the definitions can be fluid.
But there is a general distinction.
Generally, yeah. Genetic engineering is often seen as maybe adding a turbocharger to an existing car engine, modifying something that's already there. Synthetic biology is more like designing and building a whole new engine from standardized parts, maybe even parts that never existed before. Cheap DNA synthesis really enabled that build from scratch aspect.
And that building life aspect is where some of the ethical concerns come in, the playing God idea.
It's definitely a common concern you hear. The idea that creating novel organisms is fundamentally unnatural, that it crosses some kind of line. When J. Craig Venter created that first synthetic bacterial cell and filed patents, there was a big public reaction along those lines.
Is that a helpful way to frame the debate, though? Playing God?
Well, some argue it can be a bit alarmist, potentially shutting down more nuanced discussions about the real issues, like who owns these technologies, who gets access, what are the actual safety risks, how do we ensure fairness. But the underlying discomfort about fundamentally altering life is real for many people.
Which leads straight into that dual-use problem we touched on earlier.
Exactly, the double-edged sword. The same tech that lets you design a microbe to eat pollution could, in theory, be used to design a more dangerous pathogen.
Like synthesizing that polio virus back in 2002.
Or experiments that deliberately made bird flu transmissible between mammals to study pandemic potential. These highlight the risk innovations intended for good could be misused accidentally or deliberately. Bioterrorism is a serious concern.
And the whole debate around COVID-19's origins, whether it involved lab research, that plays into this too.
It does. Without taking sides on that specific issue, which is still debated, the discussion itself highlights the importance of understanding gain-of-function research, lab safety, and the potential for engineered pathogens, even if accidental, to cause immense harm. We need transparency and robust oversight.
And it's not just big labs or governments anymore, is it? With tools like CRISPR becoming easier to use.
Right, the biohacking concern. The democratization of these powerful tools means individuals or small groups outside traditional institutions could potentially experiment in ways that might be unsafe or ethically dubious. Open source biology is great for innovation, but it also makes control harder.
Okay, so biosecurity is one major cluster of concerns. What about biosafety in the environment? Releasing engineered organisms.
That's another huge one. What happens if these organisms get out? Could synthetic DNA spread and persist in ecosystems in unexpected ways? Could they outcompete native species or disrupt food webs? Think about gene drives, again, incredibly powerful, but what if they spread beyond the target population?
We just don't know the long-term effects.
Often we don't. Ecosystems are incredibly complex. Predicting all the downstream consequences is really, really hard. And simple human error is always a factor. It's often cited as the biggest risk in lab settings.
So how do we manage all this? What about regulation?
It's a massive ongoing discussion. Should we rely on strict laws, flexible guidelines, self-regulation by scientists? Existing GMO regulations often apply, but the pace of synthetic biology is so fast, there's a general agreement that oversight needs to evolve constantly.
Are countries coordinating on this?
There's dialogue. The US and China, for example, are major players and are actively discussing and developing their approaches to biosafety and biosecurity laws. International collaboration is key, but challenging.
And underlying all of this is the question of fairness, right? Equity and access.
Absolutely. Who benefits? If synthetic biology creates life-saving cures, will they be affordable for everyone who needs them or just the wealthy? How do we prevent these powerful technologies from widening existing global inequalities?
It sounds like we need a different approach to innovation itself.
Many argue for that, yes. Fostering a strong culture of responsibility throughout the research process, from the individual scientists to the funding agencies to the institutions. Ensuring proper training, strong leadership, and ethical considerations are built in from the start.
And involving the public.
Crucial. Just like the Asilomar Conference back in the 70s helped establish norms for recombinant DNA research, we need ongoing public dialogue and engagement to help guide the governance of synthetic biology. These decisions affect everyone. They shouldn't just be made behind lab doors.
So we've covered a lot of ground, from the early discoveries like the lac operon, through the development of tools like BioBricks and CRISPR.
To the incredible applications we're seeing now: impossible burgers, microbes eating plastic, CAR T cells fighting cancer, even attempts at de-extinction.
It's truly an astounding field, a testament to human ingenuity, really.
It is, but it's also important to remember it's still relatively young. There are big challenges still. Making engineered systems reliably reproducible, scaling them up effectively, predicting how they'll behave in the real world, that's all still tough.
The artisanal craft versus predictable engineering.
Kind of, yeah. Designing circuits in biology isn't yet like designing them in silicon. And as we rely more on AI, we face those black box risks, powerful but potentially opaque.
Right. So this incredible power to design and create life, it really pushes boundaries, doesn't it? What we even consider natural.
It absolutely does.
And that leaves us, I think, with a really profound question to ponder. As we get better and better at essentially writing the code of life, how do we make sure the future we're writing is, well, not just innovative, but also wise?
Yeah.
And fair.
Yeah.
And safe for everyone and for the planet itself.
Where do we draw the lines, if anywhere?
Exactly. What ethical lines should humanity collectively agree on in this new era of biological creation? It's something we all need to think about.
It's definitely a conversation that's going to shape our future, no doubt about it.
Indeed. So stay curious, stay engaged with this field, because like it or not, its impact is going to touch all of our lives.